Good morning, everyone, and welcome to the webinar on algae in human nutrition, biological active compounds, extraction techniques, and nutritional value in the face of a climate and food crisis. For those who don't know me, I'm Manuel Serrano, researcher at the Spanish Bank of Algae of the ULPGC. This webinar is organized by the Spanish Bank of Algae of the University of Las Palmas de Gran Canaria, and it's in collaboration with the Promotion Agency for Gran Canaria, within the framework of the Interreg MAC REBECA-CCT, Alicio, and the Strategic Agenda for Research and Innovation 2021-2030 of the Pan-European Platform MIRI, which includes the strategic area for research and innovation in the fields of health and food, agri-food, the environment, and energy. Before we start, I would like to remind you of a few important technical issues. Please keep your microphones and camera turned off during the seminar unless it is your turn to speak, in order to avoid any background noise. We kindly ask speakers to connect using their headphones. On the toolbar at the bottom of your screen, we have a chat, which should only be used to report any problem or error which may arise in the session's audio or video. For the Q&A session, which will take place at the end of the presentations, you can write your questions in the Q&A section. Only questions included in this section will be answered, and we thus kindly ask you not to include your questions in the chat. Please note that this event is being recorded. Therefore, we kindly ask those of you who do not wish to be recorded to please keep your cameras and microphones turned off during the entire session. I would like to thank the interveners participating in this seminar. Without further ado, we shall give the floor to a first speaker, Elena Ibáñez-Ezequiel, who is professor, a senior researcher in the Alimentomics Laboratory at the Institute of Food Science Research of the National Research Council. And today she is going to talk about a natural source of biological active compounds. The floor is over to you. First of all, thank you very much. Hello, everybody, and thank you, Manuel, for your presentation. For me, it's always a pleasure collaborating with the Spanish Bank of Algae. We've already worked together for many years, and a lot of work, and it's always a pleasure working together. So starting this seminar is a pleasure for me. I'm going to share my screen.
Una vez visto que funciona, yo creo que sí, ¿verdad? Ya lo estáis viendo. Entonces voy a iniciar. ¿Sí?
Perfecto, sí.
Vale, bueno, pues como os decía,
So, as Manuel has just said, I'm going to talk first of all as algae as a natural source of biological active compounds. So first of all, we're going to talk about algae, their importance, their characteristics, the main ones, and also whether it's possible to improve our health through algae and the relationship between algae and nutrition and food. and also finally the role they play in sustainability for our planet. When we talk about algae, why are we interested in doing research on algae? Well, we have to say that we are not really original. There's nothing new about it, because human beings have used algae for food for many, many centuries. One of the references, it could be China, where already 2700 BC, they were used as food, as well as in Greece, and the Romans. And also the Aztec as well, they were collecting algae in Lake Texcoco, especially Cyanobacteria spirulina, which is a source of proteins, which is a very important source, 60 or 70% of proteins in its composition. Nowadays, algae are really often in the media. We want to know whether they're really the food of the future, the panacea. So let's look at all these opinions. What are the characteristics of algae? They are photosynthetic organisms, have reproductive structures which are simple. There are about 25,000 to 30,000 species. They're very diverse in size and shape. And there are unicellular microscopic microalgae, and then pluricellular organisms of a big size and macroalgae. Basically, the characteristics and the main characteristics of marine algae is that they are often subject to extreme conditions during their growth, and in their growth medium, there are changes in salinity, different temperatures, water temperatures, and predators. And what algae do in order to survive is to produce secondary metabolites, which are biologically active, and they possess molecular structures which are really unique. On the other hand, given the extreme diverse biological difference, we're looking for biological products deriving from these algae is really something which doesn't seem to have any end. Algae are easy to cultivate because they grow rapidly. For most of species we have analyzed, they are easy to manipulate because the synthesis of bioactive compounds are influenced by the conditions of cultivation. Algae can be classified in different groups. We have brown algae, red algae, grey algae, and grey-blue algae. So many of them are known to us because they've been used in Asian nutrition in particular, and food. In the Western world, their use has been more limited, but they're becoming always more and more interesting as a source of biological compounds and future applications. Now, some information on brown algae. Why are they brown? Because they contain pigments, xanthophylls, which is like fucoxanthin. They also contain chlorophyll, beta-carotene, and there are polysaccharides, which are complex. And the cellular wall of these algae is made from alginic acid, alginate. And most brown algae survive in cold waters, and examples are Sargassum, Cysticeria, Undaria. Red algae, they have this color because of presence of phycoerythrin and phycocyanin. Another characteristic is their cellular wall is made of cellulose, agar, and carrageenans. The coralline alga has calcium carbonate on the surface of its cells and is used especially in bone substitution, bone marrow operations. And there are about 6,500 species of red algae, and there are also red algae such as Gracilaria, Porphyria, etc. Among the green algae, we have, for example, ulva, caulerpa, and many of them are marine and both in freshwater and seawater. Some are also on land, they grow on land, on trees and rocks. Among green-blue algae, they are also called cyanobacteria. We include spirulina. We don't know exactly whether they're bacteria or algae. Normally they used algae. They are known as algae because they use photosynthesis. And also they have filaments, which are not bacterian. Another element which is interesting, for instance, the phycocyanin give way to pigments and other compounds. Concerning the nutritional aspects, they're not well known, but they're estimated from the chemical composition and also the biochemical composition. The nutritional aspects are not well known, and the composition changes according to species, habitat, the intensity, light intensity, water temperature, all the environmental aspects for the cultivation of algae really influence somehow its chemical and biochemical composition. So in general, we can say that algae are rich in polysaccharides, in fiber, alginates, and they have vitamins, minerals, they're a source of proteins, low content in lipids, but there is presence of fatty acids, omega-3, and also rich in polyphenols. Among micronutrients, I would like to underline the presence of polyphenols, such as these. We can see here structures of this polymer, which come from this monomer. These are fluorotenins. So what are the biological activities of these compounds? We have seen that they have an impact on the detoxification of heavy metals. They have an antibacterial effect. They also, there is prevention effect on vascular risks, and also protect food against oxidative degradation, and also fight against free radicals. And other elements we have been working on, and Victor is going to talk to you a bit more about it in the next presentation. Scientifically, we have worked on carotenoids, which are in, for instance, in algae as beta-carotene, lutein, biloxanthin, which are present according to the algae we're talking about. Some biological activities, fundamentally, they have antioxidant effects. There are also some evidence of anti-cancerogenous effects. For instance, zeaxanthin may have effects against macular degeneration. And also, in general, carotenoids have a beneficial effect on chemoprevention cancer. Now, we're going to talk about how we can improve health using algae. Our concept of health is today is the idea of keeping homeostasis in the organisms and promoting thus health. There is a relationship we have found between food and health, becoming more and more evident. So between ingredients we're consuming and the benefits on our health. These ingredients we're using, which we are adding to food we're consuming, it has been demonstrated that there is bioactivity, so an important effect on health. And all this has been proved through experiments. We have seen different activities such as antioxidant, antibacterial, probiotic, antiproliferative. These are all the bioactivities which we have been subject to experiments. And in this order, we have carried out in vitro experiments, so we can select those algae which are most interesting to further research. And if all this can prove the effect on health, so we'll look at different health aspects and health effects. I'm going to talk now about different compounds which have been obtained from algae, such as polyunsaturated fatty acids, this omega-3. There are different beneficial effects on health, as we can see here. And once we've seen and studied the action mechanism, the FDA has given its authorization and recognized these benefits. And it has also been demonstrated that it can improve the efficacy of treatments against cancer. There are also polysaccharides, which are part of fiber. They also improve bowel movements. And polysaccharides derived from algae have demonstrated that there are activities such as immunomodulators, such as beta-glucans, galactans sulfates, and fucoidants as well, with different activities such as antiviral and antioxidant. And this has been proved in vitro, through in vitro experiments, such as also on sargassum. Experiments have shown significant bioactive activities. Carotenoids in general have provitamin A activity, and the most active is beta-carotene, with its antioxidant activity being the most studied. And also there are activities for the prevention of different diseases, cardiovascular, and also against macular degeneration. How can we obtain these bioactive compounds from algae? We have seen that there are a few compounds which can be interesting, such as polyunsaturated fatty acids, proteins, carbohydrates, carotenoids, polyphenols. And in our research group, all these compounds, which derive from algae, we're using a strategy, the green chemistry and sustainability strategy. So we are applying concepts related to green chemistry to improve energy recovery, and also we want to improve processes using innovative technologies such as pressurized liquid extraction. We can talk about these technologies which have advantages over conventional extraction methods. There is an improvement in the extraction methods because we're using solvents at high temperature and with important pressure so that solvents remain liquid over the whole process. The main factors influencing this extraction system is solvent composition and temperature, which increase the ability to solubilize analytes and also decreases the viscosity of the solvent. Then we also have gas-expanded liquids, which are solvents, liquids, where gas can be added, in this case CO2. And by adding CO2, the liquid expands in one phase or two phases. So we can modify different properties, such as transfer through reduced viscosity. We improve diffusivity, and also decreased interfacial tension. So with these type of solvents, we change pressure, temperature, and composition. And finally, among all these technologies of compressed fluids, we can talk about supercritical fluid extraction, which is demonstrated over the years that it's very efficient for the isolation of many molecules. So fatty acids, omega-3, carotenoids, which are interested in our research, and also tocopherols, terpenes, etc., in order to optimize all these. We can change pressure and temperature, for instance. And finally, in the last chapter would be algae and its role in sustainability on our planet. Algae can be used in human nutrition, feed, fertilizers, cosmetics, agricultural ingredients, and also for the treatment of wastewater and the production of biodiesel. They are a sustainable source of for the production of food, products used in food and non-food. They use residues from nutrients and CO2. They are very productive, and its biomass is rich in proteins and carbohydrates, lipids, and other compounds of high value added. So they also live in they're adapted to very extreme climate situations. In our last recent research, we have promoted the concept of biorefinery from algae using biomass and using different processes in order to obtain different products. And all this under the umbrella of this concept called sustainability, and there are also studies on the life cycle, on concerning economic impact and social impact as well. So all these processes using gas expanded liquids, we've used a medium-high pressure in which we can modify the polarity of the components we're going to, compounds we're going to extract, and modifying the CO2 present in our solvent. So starting from a determined biomass, we can obtain different compounds which can be of interest. And this is what we did in this particular instance and published it on Green Chemistry journal, and was based on a European project, MIRA. And we applied a biorefinery on Isocrasis galbana, and we obtained an extract with different compounds, and we obtained different products in order to make the best use of the biomass available. And finally, we can say that algae can be considered as an interesting nutritional profile for diets. Its regular consumption is also interesting and could be included in our diets. They are useful as functional food, but they're not the panacea or miracle food. And of course, there are also some particular aspects which should be taken care of and be aware of, in particular certain toxic ingredients. I would like to thank the research team which is working on this subject, and now don't know whether there are any questions now. Otherwise, I give the floor to Victor so that he can say a little bit more about the work we are carrying out in the lab. Thank you very much, Elena, for your valuable input and about all the biological active compounds from algae. And I think the participants will have questions, but we shall leave them for the end. So please do not hesitate to write your questions in the Q&A section. The next speaker is Victor Amador Luna. He's a predoctoral student. He's in the part of the Alimentaria group, Foodonomica, of the Institute of Food Science Research. And the title of his presentation is The Valorization of Algal Biomass Using Supercritical Fluid Extraction Processes. I would like to thank the organizing committee for inviting me and for giving me this opportunity of explaining the practical side of this work, compared with what we've seen previously, which was much more theoretical concerning the valorization of our algal biomass, in this case the microalgae Dunaliella salina, by means of the extraction process using supercritical fluids to carry out the extraction, mainly of the apolar compounds present in this microalga. As I have already been introduced, I will directly explain how this study has been approached, following the basic guidelines of any research. I will start with an introduction to the project and some background information related to the overall theoretical context of this work. Then I will present the objectives we set ourselves for this study, the methodology used, and finally the results, as well as certain new projects that have arisen From the results we have obtained and some lines that have opened up following this study, for which there are no results so far. By way of summary, I will show you the conclusions and the lines of research we are focusing on. Without further ado, let us turn to the theoretical context. As most people present here today might be well aware of, in recent years, healthcare has improved considerably, and we are faced with greater availability of medicinal products and healthcare for people. This has led to an increase in life expectancy over the last 100 years, largely bringing the average age of the population, and especially in the more developed countries, to well above the age we might have had just 20 or 30 years ago. The problem associated with this aging population, as well as the increase in life expectancy, is that the dependency ratio is also increasing. What is the dependency ratio? It is the percentage of people over the age of 65 who need assistance from both healthcare and certain family members in order to lead a normal life and maintain an average quality of life. This is not only a problem today, but it could also cause serious problems for the health system by 2050, when this dependency is expected to increase by 20–30%. But what are the reasons for this dependency? Dependency is mainly due to the so-called age-related chronic diseases. And all these diseases are caused by the aging of cells, and to a large extent, a significant part is due to a bad lifestyle throughout the aging process. These diseases are partly chronic, and we can only palliate their effects. So we have to live with them throughout our remaining life, and no medication can completely eliminate them. The problem of these chronic diseases is that we will have to live with them for many years to come, given also the improvement in healthcare. Our study is part of a national project that focuses on the role of certain extracts of natural origin in slowing down the symptoms of Alzheimer's disease and dementia, which are chronic diseases associated with aging and the degeneration of the cells in our brain. What is Alzheimer's disease? It is a set of symptoms that lead to the loss of certain motor skills and knowledge retention, leading to the loss of, in extreme cases, speech, the ability to move, and to be able to pick up external stimuli in a natural way. Generally, this condition, both Alzheimer's disease and pre-Alzheimer's dementia, it is a consequence of a problem associated with what is known as acetylcholine or choline cycle, because cholines are neurotransmitters that are responsible for transferring information from one neuron to the other by means of different receptors present in the membranes. These neurotransmitters are released into the intermembrane space, producing the connection in the receptor of the next neuron that induces an electrical stimulus that is conducted through the whole neuron to the next neuron, or producing the final stimulus that can be either a movement or pronouncing a word. If this cycle were functioning natural without any control, This accumulation of neurotransmitters in the intermembrane space would lead us to continually produce this nervous stimulus and lead our organism to produce tremors and, in extreme cases, what is known as Parkinson's disease. And this is why we naturally have certain enzymes, such as the enzyme cholinesterase, whose action is to break acetylcholine into an acetate and a choline, which is then reproduced into the cycle. There are different types of enzymes for each substrate. Acetylcholinesterase or butyrylcholinesterase, which are the two main enzymes that work in our neurons. Studies have shown that this process of cell degeneration and increased acetylcholinesterase activity that induces Parkinson's disease has, in addition to certain genetic characteristics, a strong environmental component and is mainly diet-related. How can we explain this? A very recent study has confirmed that diets rich in saturated fats and high-calorie diets induce alterations at both the membrane level of our neurons and within our mitochondria that can lead to poor neuronal response. It has also been confirmed that certain natural extracts such as papaya, coconut and ginkgo biloba extracts contain certain bioactive compounds that Elena mentioned earlier, which may have certain neuroprotective effects, inhibiting both the action of these enzymes and acting against other aspects related to Alzheimer's disease. In order to understand these aspects, we will first look at what a neuron looks like. The neuron process itself induces these problems. There are certain complexes known as beta-amyloid that are naturally produced in order to be able to allow the input and output of certain stimuli. The problem associated with Parkinson's disease can be explained by the fact that when what is known as the beta-amyloid cascade is activated, it generates a large accumulation of these compounds in the extreme bands of neurons, which inhibit the action of electrical stimuli and reduce neuronal response. At the same time, in the neuron itself, there are some components called microtubuli that allow the entry and exit of neurotransmitters and some nutrients that keep the neuron alive. These microtubules are stabilized thanks to a protein known as tau protein, which, due to an excess of phosphorylation, can degenerate and be modified into phosphorylated tau protein, losing its structure. We have just been talking about the stability of microtubules due to cell-derived processes. The tau protein that maintains the stability of microtubules can be phosphorylated, leading to the loss of stability. But why does this come about? According to some studies, it has been confirmed that part of the induction of these processes within the neuron is to some extent due to oxidative stress. In our cells, we naturally have a balance of free radicals, which can be oxygen, nitrogen, or sulfur radicals, and are produced naturally by the cell within the mitochondria, as well as by exogenous actions such as pollution or excess ultraviolet radiation, or by a poor lifestyle, high calorie intake, smoking, and so on. If these reactions occur naturally within our bodies, why is it that our cells do not end up being completely degraded, for example, by lipoperoxidation of the membranes or by the accumulation of mutations within the DNA?
because we also have antioxidant chemical reactive molecules called species that maintain this balance and redox equilibrium. These antioxidant species are capable of giving up an electron or two to reactive oxygen and nitrogen, or sulfur species are the ones that inhibit this action. Therefore, the exogenous introduction of these antioxidants into a diet would promote healthy cells, especially neurons. Among these species that we can introduce into a diet are those known as terpenoids, which are organic molecules formed mainly by the union of 5 carbon atoms, isoprene units, in different morphologies that have a high nutraceutical capacity. Nutraceuticals are substances of natural origin, mainly extracted from plants, where they are present in high amounts and which, according to several scientific studies, have several health benefits. Terpenoids have a great anti-inflammatory, antibiotic, and antioxidant capacity, and according to recent studies carried out by this research group, they also have a great neuroprotective capacity, inhibiting the action of these enzymes, which are highly excited in Alzheimer's disease. Among all these molecules, there are different groups: monoterpenes, sesquiterpenes, diterpenes, tetra and polyterpenes. In our diet, we can naturally assimilate up to tetraterpenes, which are the carotenoids and xanthophyls discussed by Elena. These are of great interest because they occur naturally in practically all fruit and a large number of microalgae, and are also pigments produced by these living beings as a defense against different stimuli such as ultraviolet light, high temperature, oxidative stress induced by the environment, and excess salinity. As in the case of the microalga Dunaliella salina, which we have used for this study. Dunaliella salina is a halophilic microalga growing in lakes with high salinity and has been widely studied, mainly for its antioxidant capacity as well as for the presence of a high concentration of cis and trans beta-carotene isomers. It has been extensively studied because of its defense system against changes in environmental conditions, inducing an increase in the presence of this beta-carotene with its photosynthetic complex as a defense against salt stress, increasing the concentration of these compounds of great nutraceutical interest by up to 20 times. This is why we have used Dunaliella for the next study, in which we set ourselves the objective of finding out what happens when we carry out ultra-high pressure supercritical fluid extraction with pressures above 650 bar. As a secondary objective, apart from calculating the extraction in two different pressure situations, at the optimum and in the new ultra-high pressure condition, which in our case was the maximum that our extraction system would allow. We also used the different plots of Dunaliella to see if what we observed in this pressure increase was similar to other conditions of the microalgae. We also wanted to study and compare the profile of all the terpenoids extracted, as well as other compounds of interest due to their antioxidant and neuroprotective capacity. And our ultimate goal was to see and compare the bioactivities of these different extracts. In order to do this, first of all, we carried out the extraction, which was done using supercritical fluids, in which the only solvent, in the absence of any other co-solvent that favored the reaction of polar compounds, was CO2. The CO2 was introduced into the sedimentation cell that could reach up to 2 liters, and the condition that were set were temperature, time, and CO2 flow. And with a variation only in the pressures, in such a way that we marked as optimal pressure for control the conditions that, according to previous studies, showed the highest extraction capacity, that is 400 bar, and reached ultra-high pressure conditions using pressure of 900 bar, more or less 50, because at these pressures the variation during extraction was very high. We used two plots of Dunaliella salina that we obtained from the same cultures, but only modifying the percentage of total carotenoids, one being 1% and the other 2% of total carotenoids in dry weight. These four extracts were first evaluated spectrophotometrically for total polyphenols and total carotenoids. Then they were injected into a gas chromatograph combined with mass spectrometry to obtain the profile of volatile terpenoids, and finally, by using liquid chromatography combined with mass spectrometry, we also determined the profile of carotenoids. Finally, the bioactivity capacity within these extracts was studied using a spectrophotometer to evaluate the anti-cholinesterase and anti-inflammatory activity, and with a lipoxidase inhibition and antioxidant activity assay. Specifically, anticholinergic activity was studied using acetylcholinesterase and butyrylcholinesterase inhibition assays, which are the two main enzymes responsible for the degradation of neurotransmitters. And for the measure of antioxidant capacity, we used ABTS and DPPH radicals. From this experiment, we were thus able to obtain the following results. Firstly, focusing only on the extraction efficiency, we could see that in both plots we obtained very similar and comparable results. In the case of ultra-high pressure extraction, we had slightly lower efficiency, around 25%. However, it could be seen that the extraction time was reduced by half. We only needed 1 hour to obtain all the biomass necessary, something that would be very positive for industrial process if we were to implement this extraction at an industrial level. For the profiles, we could see once again that the results obtained in samples 1 and 2 were similar, and that in this case, by carrying out the extraction at 900 bar, we increased the extraction of both chlorophyll pigments and total carotenoids, thus also improving pigment purity. As for the total polyphenols analyzed, we could see that there were no differences, except in sample 2, which showed an increase. However, as can be seen in the following graph, there were only significant differences in the case of total carotenoids, with total polyphenols showing no significant differences. As for the profile of carotenoids, something must have happened here with the image shown, because the profiles were exactly the same, with the extraction at 900 bar was always being better than at 400, allowing an increase of around 50% to even values close to 90% in the case of 9-cis beta-carotene in sample 2, and we also determined the relationship of 9-cis with all-trans beta-carotene, something that, according to previous studies carried out by this group, presented a certain correlation with the antioxidant capacity. We could see that we did indeed have roughly a 50% increase which led us to believe that their antioxidant capacity would also be increased. As for the profile of the volatile terpenoids, they were similar and comparable, and with an increase of 50 to over 95%, as in the case of neophytidine, which has increased its concentration twice. It is quite striking because thanks to this increase in the concentration of all terpenoids, we are carrying out a purification of the extract, allowing this extract to have a higher concentration of bioactive compounds. In this study, one of the peaks could not be identified due to its low concentration, and therefore we had to make new injections at higher concentrations, despite the saturation in the 2,6-methylcloxanol or beta-ionol, in order to characterize this peak and find out why there was a significant difference in the increase compared to these two plots, as no significant differences were observed for the peaks. As for the bioactivity of our extracts, focusing firstly on the anticholinergic activity, we could see that the extraction, in the case of the optimum, showed significant differences between the two plots used, something that was not seen when we extracted at 900 bar. We could also see a decrease in the values. This is positive because we are talking about a value of IC50, which is the concentration required for extract to inhibit 50% of the enzyme's activity. So the lower the concentration required, the higher the activity. It could be concluded that these extracts show a higher inhibitory bioactivity of acetylcholinesterase and butyrylcholinesterase action, but without significant differences, something that could also be due to the fact that we already start from a very high activity in the case of Dunaliella salina, and this corresponds in plot 1 at 400 bar at 58.35 micrograms per milliliter to inhibit 50% of the enzyme. We are planning to carry out further studies with slightly less bioactive matrices in order to see if this decrease becomes significant. The same applied to the anti-inflammatory capacity, where we saw an improvement in the extraction at 900 bar, but no significant difference. Where we could see a significant difference was in the inhibition of free radicals. If we look at the inhibition of the ABTS radical, which is the one used in previous studies, we could see that the increase when introducing our extracts at 900 bar had a close correlation with the increase studied with the ratio of 9-cis and trans, confirming the results obtained in previous studies. Thus, with these results, we are currently completing the study of polyphenols in order to determine the reasons for this non-significant difference in the extraction of polyphenols in Dunaliella salina 2 at 900 bar, and carry out a classification of the fatty acids present in our extracts, as there are studies that show that the presence of a high content of omega-3 fatty acids favors the fluidity of neuronal membranes, allowing better conditions and neuronal quality. Due to these results of antioxidant activity concerning free radicals, we also want to carry out assays on the inhibition of oxygen and nitrogen reactive species. Future studies that have arisen from these results include implementing this ultra-high pressure to other matrices that may have greater complexity, as well as optimizing extraction studies within this ultra-high pressure zone, as this is the first time in the world that extractions are carried out at a pressure of more than 650 bar. We also want to use these purifying extracts in encapsulation studies in order to favor terpenoid encapsulation without being limited by other compounds which may be present in the extract. Finally, I will now summarize the conclusions we have reached. First of all, the ultra-high pressure extraction of Dunaliella salina in supercritical fluid extraction shows a 28% reduction in extraction efficiency. However, the extraction time was also reduced by 50% and the concentration of total carotenoids measured spectrophotometrically increased by 8%. With regard to the carotenoid and terpenoid profiles, an increase of between 50 and 90% of all the compounds characterized was observed, which is not linked to what we obtained spectrophotometrically, possibly due to the fact that these compounds can generate certain problems when it comes to the colorimetric study due to color saturation. However, in both cases, we were able to confirm that these extracts were purified by increasing the concentration of our compounds of interest. In terms of bioactivity, we saw that in all cases there was a significant improvement except for antioxidant action, which showed significant differences and a close correlation with the cis-all trans ratio of beta-carotene, leading to an increase in assays related to antioxidant activity. Finally, ultra-high pressure extraction allowed us to obtain purified extracts between terpenoids, thus increasing the presence of the compounds in the extract despite the loss in efficiency. With this first study carried out at pressures above 650 bar, we are studying a new bioactivity optimum at ultra-high pressure. I will end my presentation here, and I'm quite happy to answer any questions. Thank you very much. Thank you very much, Victor, for explaining your work to us, and congratulations for these essays above 650 bar. From a personal point of view, I must say that I'm fascinated by supercritical fluid extraction, and it is also related to activities that I'm going to carry out at the Spanish Bank of Algae. We should talk more about it later, because within this Alisio project, we have acquired an SFE extractor. Thank you very much again, Victor, and to our public, any questions you may have, you can write them down in the Q&A section.
We're going to now give the floor to Cristina Ruano Rodriguez, who is medical doctor and professor of nutrition and public health at the University of Las Palmas de Gran Canaria, and she's also a science communicator on nutrition and dietetics on various radio and television programs. She's going to talk about this subject: can algae contribute to reduce the impact of climate change, taking into account the current dietary pattern? So I'm very happy to be here. Thank you very much. I'm going to talk about the current dietary pattern and also the current impact of algae on climate change. I'm going to share now my screen.
Creo que Víctor tiene que dejar de compartir, ¿eh? Porque si no, es que no la vemos. Yo sigo viendo la de Víctor.
Sí, yo también.
Sí, un momento, estoy aquí intentando luchar, pero no.
Arriba del todo, ¿sabes? Te tiene que aparecer
Vale, ya,
compartir.
Vale, gracias.
Ahí, vale.
Gracias, Víctor. Ok, entonces pues están viendo la mía, sí. Se ve la mía, ¿verdad? Bueno, entiendo que sí.
Sí, perfecto. Gracias.
Sí, gracias, Elena, gracias. Gracias. Bueno, como dijo Manuel...
As Manuel just mentioned, I'm a professor at the University of Las Palmas de Gran Canaria and part of the research group of nutrition and dietetics. And I'm also part of the gastronomy research group for our university, and I'm also working on science communication and scientific research on different aspects we've already been talking about. And my presentation is divided into parts. The first part has to do with this concept about healthy diet and sustainable, which is part of the recommendations for the future, and also about what we have done within our research group, which research with microalgae within the MAC BioBlue program. So our food and our diet should be nutritionally also interesting and fair and affordable as well, and respect sustainability. This aspect of food sustainability, we have different aspects. We have health, the food system, and the environmental also aspects. We have the Agenda 2030 for sustainable development goals, their objectives, and the aim is to focus on tackling hunger and also focus on food security and also better nutrition and promote sustainable agriculture, guarantee healthy life, well-being for everybody, and the availability of water and its sustainable management, and also fight against climate change. The WHO, according to its studies on 2050, for 2050, it's expected that there will be 30% more human beings on the planet. So we're going from a small world to a big world and a planet which will be too small for many people living on a planet. The current food system is not healthy, as we've just said, and it's not fair and also not affordable. Here, some data we have is that 1.3 billion people are facing food insecurity. They're not eating properly, and the main risk of morbidity and mortality in the world is very high, and over 3 billion people cannot afford a healthy diet. And the food system is also one of the most problematic. And the food production is also has to do, focuses on environmental pressures as well. So all these changes and all this situation has to do with changes we have to carry out in our diet, also for benefits on our health, and also so that they may play an important role in the resources of our planet. So there are individual diets, not only, but also the way we produce our food in an environmental friendly way. So we have to find a way to combine these two aspects. As far as a healthy diet is concerned, from an epidemiological point of view, from a scientific point of view, it's what has been mostly studied is the Mediterranean diet, which is based on an increase in the intake of vegetarian food based on vegetables. So it has to do with also as a protective effect on cardiovascular illnesses and diabetes and metabolic syndrome and obesity, cancer, and also mentally related illnesses. On the other hand, we have our Western diets, which are very rich in sugars and carbohydrate, refined ones, and other fatty acids, which is quite negative. We can see here the pyramid, the food pyramid in the Mediterranean diet. On one side, we could see concepts related to the environment. So we have the optimum consumption, which has an impact on our health. And then we also have the environmental pressure of the food we consume. And in general, there are different aspects we have to take into account. So there is this concept of the planet and the protection of our planet. So what are those sustainable diets? They have a low environmental impact. They contribute to food safety and also our nutrition. And in studies, and this one in particular, 2020, carried out by Fresan and other researchers, has shown that foods which contribute mostly to sustainability, which have a higher pressure on our environment, is the consumption of red meat, of processed meat, of dairy products, and fish. So these four types of food have a large impact on our environment. Especially processed meat are not beneficial for our health. Fish, we have omega-3 we've been talking about, and there are also aspects, negative aspects of dairy products. And there are diets which are very rich in these elements, are not sustainable. This is a study carried out by the Sun group in Navarra, in Spain. And there are types of foods which have a higher impact on the environment: is processed food, processed meat, and dairy products and eggs as well. So all these food is gives us different elements for the good functioning of our body. We have different models here according to the greenhouse gas emissions. Here there is a comparison in the Netherlands, and diets. We're comparing vegetarian, vegan, and Mediterranean diets, and the vegan diet is the one which has the least impact in terms of GHG. And following the same models, dietary models, diets which have a high percentage of meat also have a larger impact. And we take into account the GHG emissions is mostly red meat and cheese are the ones which have a higher emission of GHG, and it also has a larger water footprint on water. And we can see beef, for instance, or legumes, pulses, and soya or broccoli. So given all this background, the recommendations would be to have a diet with more vegetables and less meat. And how can algae contribute to this situation? And how can we introduce them in our diet? Because they could be a sustainable source of food and can also contribute with omega-3 and proteins. Dr. Ibáñez already talked about it, but we can see here macroalgae and also microalgae. Macroalgae, also called seaweeds. We have different types, brown, red, and green algae, and different compounds like vitamins, minerals, dietary fiber, proteins, as well as antioxidants. So all these proteins are good quality. These are in, we can see the reference here is Dr. Pinchetti, and we have here a list of algae, whether they are brown, green, or red, and their chemical composition in general, large amount of carbohydrates and fibers in particular, and also high consumption as well as far as proteins are concerned. As Dr. Ibáñez just mentioned, they're high quality proteins, readily available. They contain essential amino acids which cannot synthesize normally, and so this would allow us to eliminate meat, eggs, etc. from our diet and dairy products. We should also look at the availability of this protein, because there are not many studies from a nutritional point of view, not many experiments, clinical experiments, which can really confirm that we can substitute one type of food by another, and that algae could introduce and make this a change in protein intake. Above all, we use algae as thickening, gelling, emulsifying, and stabilizing agents. They have especially this thickening and also gelling, also especially in Western diet. I would like to say that fiber is mainly soluble, so it can help us to regulate the level of cholesterol in our blood. And also it's a source of food for microbiota, and our research, which has been carried out, has shown that bacteria are essential for health.
Bien, nosotros hicimos
We
una revisión
made a
sistemática
systematic
sobre el uso
review
de
of
macro y
the use of micro and
microalgas
macroalgae
diferentes componentes
in
del síndrome metabólico,
different
como
elements
fueron la
which
obesidad,
concern metabolic syndrome, like
control,
obesity,
obesidad,
like
hipertensión,
hypertension,
diabetes tipo 2 y
type 2
dislipidemia.
diabetes, and
Realizamos,
dyslipidemia.
ensayos
And
clínicos
we carried
randomizados que
out
hubiesen trabajado
clinical
con algas
tests
o
using
macroalgas, sobre todo,
seaweed
también microalgas,
and
y
also
también
microalgae, and
pigmentos
also antioxidant
antioxidantes,
pigments like
ficosantina, que ya les comentaba
ficosantine
anteriormente, y que son los que tienen
and
un gran interés también sobre la
other
salud.
elements which are of interest for our
Lo
health.
que vimos fundamentalmente es que hay una gran ausencia. Encontramos 12 artículos de
We found 12 papers
del estudio
within the MAC Blue study.
Pero la realidad es que no hay tanto
We have seen there are not many clinical tests which have been carried out to all these beneficial effects.
Bueno, ahora pasando a las
And concerning microalgae,
las microalgas tienen un gran potencial también.
they're also
Las microalgas como
very interesting from this perspective, from different effects, as Victor just mentioned. They're working on Donariella salina, though there are many possibilities for the future concerning their beneficial effects on health. But the problem is that many of them are not catalogued for human consumption. So there is a whole procedure, this novel food procedure, which is very costly and complicated. Legislation and this potential can be, however, developed in the future. And chlorella and spirulina have already demonstrated to be very beneficial for our health, particularly for the antioxidant pigments. We also carried out a study, systematic review on Athrospira, which is known commercially as spirulina, which is a microalga which is being mostly commercialized, which is being used nowadays. And the study was published in the Journal of Applied Phycology. And we also only made a revision of a randomized study, and there are many clinical experiments which don't have which are scientifically, from a scientific perspective, are not that valuable, unfortunately, because they have with some problems in their realization. However, we included 50 randomized so they were used as nutraceutical, not as food. And we wanted to see its antioxidant role and effect. So we could see this were benefits on dyslipidemia, on hypertension, on obesity, and also on immune response. So everything has to do with this antioxidant aspect. So all these studies Dr. Ibanez was talking about, minimum is 3 grams per day, which should be the normal intake to obtain any benefit. And the important thing would be to define exactly what the doses could be, and also should I just use it regularly or just once a week? This also has to be determined. So we also analyzed chlorella, So we have much more evidence on Anthrospira compared with chlorella. And the main value of consuming Anthrospira and chlorella lies in its antioxidant profile. And the important thing, this concerns also the cholesterol regulation and the regulation on hypertension and glycemia in blood and on body weight. But longer studies should be carried out with more participants and with better criteria and qualitative criteria in order to establish which dose should be recommended for the population as intake. These are the studies peer-reviewed for qualitative criteria and beneficials on health, and we could see this in our systematic reviews. And finally, within our institute, the University of Las Palmas, both our group and endocrinology group, we are coordinating ourselves within the MAC project, which is financed, MAC-financed European project, and we participate with the Canarian Technological Institute of Canary Islands and Spanish Bank of Algae, and we're trying to see if we can obtain new products from the biotechnology of the Macaronesian region. So how they could be used as in human nutrition or as feed for fish, and how we could thus enrich feed and then later food as part of the food chain. And we, in our research, we carried out with mice on Donayella salina and Tetraselmis. We used both microalgae as a natural source of food and sustainable, and we used a control group with also experiments with mice, randomized, and we had 18 mice, and they were fed a diet rich in lipids, and we also introduced on one hand Donayella and on the other Tetraselmis to see whether to see the results as far as the metabolic syndrome is concerned, and also glycemia. They are very simple experiments. And we couldn't see any significant effect from this intervention, not even on body composition or weight, and in both groups, control group and intervention group. So there were significant differences in water consumption and in the weight in the first 2 weeks when they were consuming particularly Donayella salina. Very basic experiments, but we would like to continue along this line. And to finalize the content in fatty acid content, as Victor was mentioning, is the importance of omega-3 on health. It's something which has been studied already for many years. Its influence on health And we could see that microalgae can be an important resource also for fish and from fish or krill, and they could be the main sources. Population is not taking right now as cardiovascular prevention, a lot of omega-3 fatty acids. In the Canaries as islands, despite this, we are consuming always less fish. So we might consider microalgae as an interesting source of additional nutritional elements for the population, because the fatty acid percentage is much higher, especially this composition in interesting fatty acids. And the composition, the different species which have been studied, we have looked at higher accumulation of DHA and compared with EPA. So we have seen that the accumulation has been much more interesting as far as phospholipids is concerned. So it's an interesting source of omega-3 DHA acids. Omega-3 should be in bigger quantities compared with omega-6 in our diets. Here we have another study, omega-3 fatty acids, EPA and DHA, whole health benefits throughout life. Very interesting as far as the cognitive function in fetus and the immune system, also cardiovascular effects on inflammation and on different aspects of our health. And also as far as prevention is concerned in weight control and cognitive function and Alzheimer. These are some examples of companies which are using microalgae, such as phytoplankton marino, and also here the ITC, there is a spin-off called Alimentación. I think that Antera or somebody else from the team can comment more on this, but Alimentación also produces microalgae, and the Spanish Bank of Algae is the main bank for algae in Europe. So the microalgae which we can use are gourmet products. It's not something that an element which, even if beneficial for our health, can be recommended for the population in general. The problem often is that many products are not affordable for the population, and also very often they buy low quality products. The idea would be the idea of introducing algae in our diet could be a sustainable and also affordable and fair element in our future diets. Thank you very much again for inviting me, and I'm available to answer any questions. Thank you very much, Christina. Thank you very much to all the three speakers for sharing with us your point of view and your contribution. And now we are going to show you a video and to show you the objectives and results of the Alicia project, which is closely related to the topics that we have been discussing today. So we can now look at the video. What should we do?
Alicio, un proyecto de investigación e infraestructura.
Alicio is a research and infrastructure project that aims to strengthen the operation and quality of the services of the Spanish Bank of Algae,
El único biobanco de algas reconocido a nivel estatal que desarrolla
which is
su
the only
actividad
algae
de I+D+I
biobank recognized at
en el
national
campo de la
level that
biotecnología de
develop
algas.
its R&D activity in the field of algae biotechnology.
Procede del Fondo Europeo de
It is
Desarrollo
financed by
Regional,
the
FEDER,
European Regional Development Fund and from funds from the government of the Canary Islands, which has allowed the acquisition of high-tech equipment and the hiring of a multidisciplinary team of experts in different areas with the aim of improving the supply and quality of genetic resources. The molecular biology department has hired a researcher responsible for the extraction and purification of nucleic acids and proteins, as well as assays and analysis of biological samples. His experience in identification techniques allows us to implement improvements in the molecular unit and update the database for taxonomic, phytogenetic, genomics, and biotechnological purposes of the microalgae and cyanobacteria collection of the Spanish Bank of Algae. In the biotechnology unit, a doctor in biochemistry has been incorporated whose objective is setting up and implementing the equipment obtained through the project funds, such as rotary evaporator, which is used for the elimination of solvents in biological sample extracts. The Alicio project has provided us with a gas chromatograph used to determine the fatty acid profile of the cultivated strains, and a digestion apparatus for the determination of proteins from macroalgae, microalgae, and cyanobacteria from the BEA collection, providing us with valuable information on the nutritional quality of the species. Our biotechnology department has also been reinforced with the hiring of a chemical engineer that allows us to maximize production systems by implementing improvements in the experimental cultivation plan, processing the biomass, minimizing production costs, and the environmental impact of our activities. Another equipment acquired for the project is a freeze dryer, which is used for dehydration of biological samples while maintaining the quality of the biomass produced, as well as an extractor with supercritical fluids, which allows the preparation of eco-sustainable extracts and metabolites from biomass. In order to manage the project and transfer knowledge and results to entities working in the blue biotechnology sector, a project manager has been hired who is in charge of dynamizing and being the link with companies interested in using these results. This allows us to effectively and correctly develop the aforementioned objectives, create collaboration and transfer agreements, generate the first activities and projects to identify strengths and weaknesses in ideas, processes, and products. Thanks to the staff and equipment which has been successfully and efficiently added to the already established workflow, the BEA has established itself as a reference in the European Initiative for Algae and favoring the diversification of the regional industry in order to improve the state of the local economy.
Bueno, como se ha podido ver en el vídeo, tanto el proyecto REBECA-CCT como
As we could see, be seen in the video, both the REBECA-CCT project and the Alicio are closely related, as REBECA-CCT allows us to continue increasing the collection of strains from Micronesia and also creates a synergy with Alicio, facilitating in-depth studies of the current and future biological industrial applications of collection of macroalgae, microalgae, and cyanobacteria developed by the Spanish Bank of Algae. We are a bit behind. We invite everyone to ask questions to the speakers using the Q&A section. We already have some that were collected via the registration form, and we made a selection, and some of them already talked about during the presentations. However, if there's any other question which we will not be able to answer, you can always send them by email at the address info@marinebiotechnology.org. I'm going to introduce you to the scientific directors of the Spanish Bank of Algas, Juan Luis Gómez Pinchetti and Antera Martel Quintana, the curator of the Spanish Bank of Algae. The first question: do we have any lists of cultivated seaweeds or seaweeds that can be cultivated that are being considered for both human and animal feed? Don't know whether Antera or Juan Luis would like to answer this question. Don't know whether they're available. Thank you. Thank you very much. Thank everybody. Many ideas have been raised and many things to comment on. So this question currently, as far as the algae which can be used and in diets within the novel food regulations, they belong to the novel food catalog. And particularly in Europe, there are some species which are recognized and which are used from a traditional point of view in Europe. And concerning this, nowadays, more than 90% of the biomass deriving from algae in the world come from cultivation in open and closed systems, and others are also limited to industrial applications such as phycocolloids, etc. So most species in algae which are consumed nowadays derive from cultivated areas and under controlled conditions. And many of these species can also be used in feed, and there are new species which we have been characterizing, and for instance, Asparagopsis, for instance, this genera can be cultivated and also can be used in feed for different animals, cows in particular, and to reduce the production of the emission of GHG gases. And so there is a mitigation possibility in this sense. Thank you very much, Juan Luis. There is another question. The protein phycocyanin in spirulina has been found to have high antioxidant properties. What would be the good extraction method, probable extraction method for this? Could it be incorporated into the food sector? I don't know whether Elena or Victor could answer this question.
Vale. Bueno, sí. Realmente
in this case, we should try to preserve the original structure of the protein. We have studied the extraction of different proteins from spirulina, and we've done it with high-pressure systems and using as water solvent and at low temperature, so that we can keep the structure of the protein, and we are favoring and the extraction using high pressure. But basically, we're extracting it using water.
La siguiente pregunta, ¿podría llegar un futuro en el que pudiésemos cultivar algas en casa para autoconsumo?
Could we in the future be able to grow algae at home for self-consumption?
¿No les importa? Porque yo mis inicios,
I can answer this question because when I started in biotechnology, I cultivated spirulina at home. I bought a metacrylate tube and all the technique necessary for this. So I tried myself, and for a family, then it should be maybe maybe the equipment should be adjusted. But there are ways to organize and develop these types of activities, I think. The same for other types of food. And now we go on to the next question. Is there any limitation in the consumption of algae for the excess of in any nutrient? And then there is another one in the chat.
La otra dice, ¿será sostenible una alimentación basada en algas teniendo en cuenta que un consumo recurrente de ellas actualmente no es del todo saludable?
Will the use of algae be sustainable, bearing in mind that the recurrent consumption of algae is currently not entirely healthy?
Y luego hay otra pregunta.
Then there was another one.
Yo creo que es la segunda, ¿verdad? La de,
sí, la de Paula Fajardo, que te da las gracias.
Paula Fajardo, I wanted to thank you for the presentation, and I don't know whether you wanted to answer this question directly, Elena, or Cristina can also give her point of view.
Pues yo creo que Cristina, yo creo que te toca Cristina.
I can think that Cristina, as far as different diets are concerned and nutrition, you can answer this question. You're the expert, Elena, and also Victor, as far as the consumption. I think that algae can be really sustainable and healthy. And what I didn't really you talked about different questions, but they're all related to the consumption and the safety.
De implementar o la seguridad que podríamos o qué observación futura podríamos tener para implementar las algas en nuestra alimentación.
How could we include algae in our diet? Are there any negative aspects? I think it's the excess in iodine. I think Victor can talk more about it. Especially when we introduce new food in our diet, as far as algae are concerned, some of them have a high iodine content, so it's important if you don't have thyroid problems in particular. If you have a medical problem, of course, you have to go to the doctor. Health can be improved through a diet, but we are not generating drugs. We are here to preserve good health or to improve health, but not to cure specific illnesses. So if somebody has a thyroid problem, should not consume high iodine content. What Cristina just said, one of the most important problems we have been faced with is different sources of proteins, and this is why algae can really contribute as a substitute for proteins from meat, and the same for insects as well. These proteins could also be interesting, and algae are another source. And as far as heavy metal content is concerned, in order to be able to consume, there are controls which algae are subject to, and one of the parameters to be taken into account is the evaluation, measuring of their content in algae, because they could be accumulated and they could become toxic because of this accumulation of heavy metals. So it's the same as for other food, types of food, in order to guarantee safety. I don't know whether, Cristina, if you want to add anything else. If one has any problems, medical problems, you can see the doctor, but otherwise there are no miracle food. We're in a context of a healthy diet, and algae can be part of it, but they're not miraculous.
Y es un error, ¿no? Entonces también hay que ver la dosificación, porque a lo mejor no es tanto el yodo, no nos va a perjudicar tanto.
Iodine will not possibly be a problem, because it also depends on the way they are cultivated and the species. So it's really recommendations for the general population, these healthy recommendations we can give. But then very specific questions, we should talk about pharmacological aspects and not nutritional aspects. And with the inclusion of algae in our diet, we could see in our studies that they could be used for the prevention of some illnesses, and we need more clinical studies. More scientific evidence are necessary in order to be able to define specific beneficial aspects of algae and for specific conditions. So these studies should be carried out further. So it's a long way we have ahead. Thank you very much, Elena. First of all, José Luis, we just wanted to say, Victor is going to say, I just wanted to stress this concerning heavy metals, as we just said, all macroalgae, which are consumed nowadays. They are being cultivated under controlled conditions, and they are really checked. All this elimination of heavy metals is carefully checked. So all these nutrients, which excess nutrients, also we continue with in vitro experiments and cultivation. It's possible to evaluate the best or maximum daily intake. So they are quite controlled. Very quickly, I just want to remind that iodine and heavy metals, not all algae and not all species, micro and macroalgae, are for human consumption. We cannot consume them all. We have, we're now studying those several which have to do with our diets, but the iodine is, this subject has more to do with brown algae. This negative publicity, which has to do with kombu or wakame, is excessive. So concerning heavy metals, not all algae can be consumed, and most of them which are being consumed today, they are grown in controlled conditions. So the cultivation medium, but also in the open sea, through aquaculture, and in hatcheries, All this is controlled, and we have regulations which has to be complied with. And for instance, in Japan or the United States, it's and as well as in Europe, there's a series of whole legislation on edible algae species. And we should never forget that there is a specific interest in Europe in the development of production and the applications derived from microalgae, and they follow a specific regulations. And also, we're developing standards for algae and algae-derived products, which we'll have to comply with, and which will be a reference for further knowledge which will be generated in this field. Thank you. I'm very happy that you could all participate, because it's a very common question when we talk about algae in human nutrition and diets. So now we go on with the next question. Have algae been used in feed, in aquaculture? Algae are used very often, and as and also for beef production, and 2% of paradopsis in animal feed, also for cows and beef animals, to reduce the methane they produce, which is 15%, represent 50% of GHG at a global level. The next question. Is it possible to grow algae in axenic conditions in large bioreactors? I don't know whether Ante would like to answer this question. Thank you very much. Very happy to be attending this webinar, and it's possible to cultivate axenic algae using carbonic nutrients without parasites, and algae can be acquired axenic or with a reasonable quantity of bacteria, some especially bacteria which are not pathogenic. So it's possible in closed systems. It's possible to cultivate axenic algae in these systems.
No te lo oímos, Manuel.
Thank you very much, Antera. There was another question. Will there be a certificate of attendance? If you ask for it, of course, could be sent to you. You can write to us at info@marinebiotechnology.org. The methods of extraction methods of bioproducts and their characteristics and all applications, will they be the same for microalgae? Are the extraction processes similar to other processes which are not related to algae? Is that the question? I understand that the extraction methods are the same for different bioproducts which can be obtained. The extraction techniques depend on the process you're going to carry out. If you want to use different types of this extraction with pressure fluids. It depends on the extraction we made. In FSC only uses CO2. The extraction with supercritical fluids using CO2 has to do with apolar fluids, and so we should include probably a different solvent in other situations, such as methanol, ethanol, and we're also studying introduction of NAVIS, which are solvents, synthetic solvents, which also favor the increase, the extraction potential of these systems. So it depends a little bit on what you want to obtain. You will use either one or the other process in order to favor the obtention of certain compounds. So we try to mix and use biorefinery to extract as much as possible and then analyze its potential. Thank you very much, Victor. Another question. When is it estimated a part of the population will include algae in their diet? I would like to answer this question, and I think that it can largely be due to the acceptance by the consumer and the how far the process can be economically viable, and also the, let's say, democratic support for these activities, whether it can be on a state level or if anybody else would like to add anything to this, please raise your hand. Elena would like to say something. I would like to talk about one aspect mentioned by Christina, which has to do with obtaining algae or cultivations which are accessible so that people can really affordable. So if it's too expensive, it cannot remain just a nutritional element and just for few people. It should also be interesting from an economic point of view. For instance, biorefinery is an important element, an aspect, so we could reduce the costs associated with the production of algae, so we could probably valorize products derived from algae, and so reduce the end price of algae. But until algae remain so expensive, it's difficult for them to be used in the wider population. Any other question? This question is quite interesting. How does Spain compare with other countries in terms of algae production and their value-added byproducts? Is it ahead of other countries? Is it behind? Anyone who would like to? Juan Luis can answer. We have some reports. We have received recently some papers, academic papers, which refers to European maritime aspects at European level. And so companies working in the sector, with both macro and microalgae, and particularly in Spain, the last statistics available show that we are the top in the list of the companies producing algae, with other countries being Ireland, France, and Portugal. And as far as Germany is concerned, for microalgae, also Germany, Italy, European countries where much more production is being now promoted for these products. But Spain is high in the ranking of these companies, and for macroalgae, there is an activity which has to do with the industry producing agar and alginates. So a lot has to do with these fisheries as well in the north of Spain, and we're really at the top in the European ranking as far as Spain as a country and algae is concerned. Thank you very much. Looking at different questions and the Q&A, there are many aspects to comment about. I think there was one which was for Victor from José Regidor, which has to do with its reference. I think he wanted to participate.
Sí, yo entiendo que el profesor José Regidor quiere hacer un comentario, a ver si se le puede dar.
Sí, correcto.
Sí, ¿se me oye?
Can you hear me?
Sí.
Sí, perfecto.
Yes, we do.
Muchas gracias.
Thank you very much. First of all, I wanted to say that I don't belong to the world of chemistry or algae, but I'm very much and emotionally linked to the Spanish Bank of Algae, and this is why I'm here today. And the comment I wanted to make is something I would like to say, that I'm a neurobiologist, and I'm specialized in processes due to neurodegeneration and Alzheimer, and it's such a complicated subject, and it's difficult to have subjects and examples which can complicate the view of the situation. For instance, the acetylcholine is an element, a secondary element within the Alzheimer condition, and avoiding acetylcholine, which is negative for the for our brain. These are drugs we're actually using. It's not, acetylcholine is not the real problem within the brain. So just wanted to say to be careful with complex elements and aspects that even for us specialists is very difficult to determine. I wanted to say thank you for your comment. I think it's a very complex condition, Alzheimer, but I wanted to say that these are three particular activities, and beta amyloid and the other protein. There are three consequences and a secondary consequence derived from Alzheimer or influencing Alzheimer, but could be prevented somehow through the algae consumption. But there are so many aspects which cannot be treated through a change in diet. It's a very complex condition. You are quite right in using and talking about these aspects. It's just being very careful in using different aspects, especially in diet and metaboloids and different proteins and tau. Protein, there is another neurotransmitter, which is glutamate. It's quite complicated and very difficult. So it's better just to maybe give examples in other areas of knowledge. Thank you very much. And also I wanted to another question. And when will we have less technical and administrative and bureaucratic hurdles will be solved so that we may really cultivate algae in the Canaries? I think it's an excellent question. Well, know where you're coming from, and I thank you. I think that Professor Regidor knows what the answer is. We've been working on it for many, many years. How can we develop activities in the sector of chemistry and algae in the algae, because we have such a perfect and wonderful climate for this kind of cultivation. And we are working on a platform where we could detect bottlenecks, a whole series of bottlenecks which have to do with regulatory aspects. And it's a question of informing and working on how we could improve legislation which has to do with agriculture, especially animal agriculture. And we're working on them. And all this effort we are making, and over the few years, I think we have examples, very successful ones, and we hope that our different companies can really develop their activities through our research. Thank you very much. Thank you very much. I think this session is really very interesting, this Q&A, but we have to close the session today. And any question you may have, you can send to info@marinebiotechnology.org. And you can also find this address on the webpage, on the website of the Spanish Bank of Algae. Also, you will also receive, all participants will receive a recording of this session. And I would like to thank Elena, Victor, Cristina for their availability, presentations, and for answering all the questions set by the participants, by the team. And also thank Artemis Rivero from the FPCT for his technical organization, support, promotion, and all the partners of the REBECA-CCT project, the Society for Economic Promotion of Gran Canaria, to the colleagues of the Alicia project, and to the rest of the BEA colleagues, and especially all the participants for their interest shown and participation and the response given to this seminar on algae in human nutrition, biologically active compounds, extraction techniques, and the nutritional value in the face of a climate and food crisis. We hope that you have enjoyed today's seminar. We shall say bye for now, and we will keep you posted through the FPCT website and our social networks about the next REBECA-CCT webinar. Thank you very much to you all.
Recording stop.